Now I'm thrilled to introduce Dr. Laura Smoliar, the Executive Director of the Taiwan Science and Technology Hub at Stanford University. She is one of our LDV capital experts. Laura will moderate the panel on visual tech advancements in life sciences and biotech. Laura, it's great to see you, and feel free to take it away.
Thank you, Ash. It's wonderful to be here. We have a great panel lined up here. The explosion of AI coupled with visual technology is a huge unlock for the life sciences. Training models on multimodal data is powerful and enabling. We have three leaders pushing the boundaries of overcoming complex genetic diseases, improving health span, and creating actionable insights into environmental impacts on health. Professors Olga Troyanskaya and Elizabeth Neumann are at Princeton University and the University of California, Davis, respectively. And Neil Inala is the CEO of Therapeutics. And now I'll ask each of you to introduce yourselves briefly, starting with Neil.
Thanks, Laura. Hi, everyone. It's very nice to see everyone, and thanks to the LDV for setting up this summit. I am the CEO of Therapeutics. Nobias is a recent biotech startup specializing in taking a very large genomic database that we have access to through a hospital partner. It's the largest pediatric genomic database in the world. We take that database and apply modern AI techniques to that to extract a stream of biological insights, which we use to develop new therapeutics, new drugs, and basically new molecules. We are a clinical stage biotechnology company. We have recently completed a phase 2 study in a rare pediatric indication, and we are developing a broad and interesting pipeline in a number of therapeutic areas.
Thank you, Neil. Olga, can you introduce yourself?
It's a pleasure to be here. I'm Olga Troyanskaya. I'm a professor of computer science and genomics at Princeton University. I'm also the director of precision health at Princeton and the deputy director for genomics at the Flatiron Institute of the Simons Foundation. My group's goal is to use AI and data science as the integrator to transform human health. And we're hoping to really do this by integrating this across all levels, connecting genomic to social to lifestyle and environmental, with the goal of enacting smart and equitable clinical practice and health policy.
And Elizabeth.
Hi, I'm Elizabeth Neumann. I'm an assistant professor at UC Davis. I'm in the chemistry department. I'm an analytical chemist by training. So I focus on how do we make a measurement? And so I specialize in mass spectrometry, but I realized really early on in my career that focusing on one instrument and one omics space is actually doesn't tell you the whole story. And so we really run the gambit from genomics all the way to secondary metabolism in an imaging context for crazy systems like undecalcified bone and bone marrow, all the way to spinal cord and whole body systems. And so we really run the imaging and spatial biology field from the chemistry perspective.
Great, thank you. So I'm going to ask each of you to talk about how the evolution of visual technology and its intersection with machine learning and AI has impacted the work in your field in the last 3 years, the last 12 months, and what you see as problems that are feasible to tackle now that just weren't possible before. And also, why is spatial biology garnering so much attention now? So I'll start with Elizabeth, because I know that is your passion there.
Spatial biology is really cool, and I say that as a chemist. So, it's got to be at least moderately cool. The chemist is on board. And so machine learning is neat. So if you think about mass spectrometry, we're detecting tens of thousands of features in fractions of a second. So we can probe 30 cells a second. And so as you can imagine, that's tens of thousands of things we know and about hundreds of thousands of chemicals that we're detecting that we don't know. And so using machine learning, it allows us to really quickly parse through these different molecules that we're detecting and then see how they actually are intrinsic to either a cell type or a disease state. So it allows us to look at these large patient cohorts very quickly, running the gambit from individual cells all the way to the full organ system very quickly. And so that's evolved since I've even been a graduate student, our ability to do this and how much data we can actually handle. It's quite impressive.
Thank you. Well, Olga, what's changed over the years for you?
So for us, of course, my group actually builds AI frameworks to decode genomes and understand cellular specificity and how your genome is related to health, to be able to detangle the biology underlying complex diseases. So for us, really, basically the AI revolution has been really transformative because the emergence of deep learning approaches really has enabled us to pioneer the use of these models to study regulatory genomes. To be able to computationally predict what any mutation in your genome, especially those outside of genes, which is 98% of your genome, that actually control how genes are turned on and off, will affect your cells, organs, and of course, finally, your health or treatment from disease. So of course, these don't act in vacuum, they act in specific cellular and tissue and disease contexts. And these, the explosion of multi-omic and spatial data are critical here to be able to really understand these effects of these mutations and of your genome sequence on your health and disease. And so we're now able to really start developing AI-based integrative approaches that are able to study the genome, especially the non-coding genome, and model these complex molecular level changes captured via diverse multi-omics techniques, including in specific clinical treatment, environmental contexts. So that really can enable us to really have a systems level molecular view of human health and disease.
Wonderful, thank you. And Neil?
Yeah, so as a drug discovery company, our general problem and our biggest problem is really understanding how can we turn this huge variety of information, giant databases, disconnected databases in many cases, into something that's useful and actionable for really drug development. There's a secondary problem of like, how do you develop a drug, a molecule really, that will make it through this sort of series of gauntlets of, of course, just being effective, getting through the liver, getting through to the point of application in the cell or within at the target site, and then of course getting it through the FDA and showing efficacy and so on. But the core to the problem we have is, how can we know more about what's out there? How can we turn these disconnected databases into something that's useful, really? And that really comes from developing greater tools for understanding. I read something once that says that the information, all of the sort of data streams that we have in the world go through several levels of transformation, where they start as data, which is just the raw coding of things, and then turns into information, which really is you start to add a little bit more structure to things. And then we have knowledge and eventually, hopefully, wisdom. Now, that process of turning information into knowledge and wisdom is a little bit mysterious, actually. We still don't really know what it means to understand something. I think there have been a few huge breakthroughs, though, that get us closer to that. Development of the concept of embeddings and latent space is a big one. I remember, when I first got interested in AI, like a long time ago, I think I read a Dr. Dobbs journal on artificial neural networks in like 87, something like that. And when I saw that, I built a program to play Connect 4, the the children's game. And I remember it being very difficult to encode the loss function to really play a two-pronged attack, because otherwise it's too easy to beat the computer. And at the time, it was a lot of just like tweaking random things and understanding like, how do these things kind of connect and what's the math behind this. But I wouldn't say it was knowledge and wisdom, right? Whereas now, with a lot of the discoveries that have come through, particularly in the space of like semantic and instance segmentation and visual programs, and also like with generative AI, I think we've seen a much greater expansion of the tools that exist that might enable us to understand things a little bit more. And once we understand things, we can pull together sort of disparate connected concepts. We have a much greater chance of building something for drug development, for example, that will actually be a useful molecule rather than just like trying a whole bunch of random things and hoping that it works.
Thank you. For my next question, and by the way, if you have questions for the panelists, if you put them in the chat, I think I should see them so I can try to field those. What are the most exciting developments on the horizon from your vantage points, and what tools and or resources are most enabling for you? And is it an access or a challenge to get these tools in academia or in a startup as opposed to, the large tech companies? And how are you addressing that? I'll start with Olga.
So it's very exciting for us, of course, between the explosion in the AI space and, of course, the explosion of multi-omic data. I think that really, to me, the most exciting promise is really the genome-based integrative approaches that can really change how we do precision health. I think that, it really does rely on a lot of compute, which, of course, while I'm not, I'm affiliated with both Princeton University and the Simons Foundation's Flatiron Institute. So I'm in a very lucky position for those outside of the really giant tech companies. So we do have quite a bit of computing resources. But that said, we're never actually quite at the level of what we need. And of course, if you talk about startups, I'm sure this is a much more limiting question as well. And of course, I think being able to really leverage this to identify the genomic signals that can be combined, both for diagnosis and for treatment guidance, can really enable the future of treatments that are just really coming on the horizon. Honestly, I'm most excited sort of in the health space that all of this tech revolution and data revolution can really enable us to start looking at treatments that don't just modify proteins, but really are targeting the regulation, targeting how expression is regulated and which version of the protein, right? Like which isoform is actually being produced, targeting RNA. And I think that there's enormous promise there to be able to really be able to reach the previously undruggable targets, both proteins and pathways, as well as potentially providing much more precise control for these treatments and minimize side effects. And of course, that relies on continuous access to data, right? Having data as open as possible. So that's another side of this, right? In terms of the resources, as well as continuous ability to develop novel AI approaches and actually be able to run them with enough compute.
Thank you. And Elizabeth.
As an assistant professor, we're like science poor. So like, got to get creative with how we can access tools and like how we actually it's very difficult to buy nice computers on like NIH grants, right? And so we're at the mercy of the ability to get creative with how we actually access these tools and then how we can afford them. So sometimes, and then also how we explain them. So like I work with students primarily. And so at UC Davis, we are a Hispanic serving institution and 60% of our student population are first generation. So I have graduate students that actually didn't have a computer in their house, right? And so now I'm having to teach them not only how to do mass spectrometry, but now how do you analyze, 3 terabytes of data with something you've never used before? And so when you think about access to these things, we have to use the cutting edge. Our data is massive and quite complex. And since we're spanning multiple omic spaces and then multiple spatial scales, how do you actually train a student who's never really had a computer before to do that and do it quickly and then get a paper in 6 months and then be able to explain it for their qualifying exam? And then separately speaking, I was on the phone with someone yesterday and they were like, I've spent 6 months trying to get this one software to work. I spent 5 minutes on the phone with them and they got it. And it's some of this translation to, I think, the general population. They're a physician. So like very much out of the AI field, but very critical for how we understand health. So how do you actually translate these really cool approaches to like a more general audience, or how do you make it available and accessible to things like students, right? Because it's my students that are going to be the people working at these or starting the small startups, right? Or working at the startups or working at these companies. And so the ability to get them trained, and I think we're talking about this later, is I think critical for actually the future of both using the technology that's available, but actually making use of it in really clinical context.
Oh, thank you. Yeah. Neil, what do you
Yeah. So I think that, for us, the most exciting things that are coming down the pipeline are probably things that stem from, a greater understanding of, tools to organize and develop information and incorporate that into something that's useful for us. That really, there's not like a general answer to that, but like specific things are in generative AI, for example, which most of you probably know from Stable Diffusion, Midjourney, things like that, where you maybe you type in a text that describes an image. I think one of the previous panelists was talking about this, approach for video generation and so on. But type in text that describes an image and then it generates that image or a video or something like that. The general concept behind that is basically exploration of noise generation from a semantic space. And we use that concept for a variety of things. For example, for generating conformers, which are used in, efficiently sampling which kinds of molecular setups or conformers are able to interact with a protein. But more generally, I think data gathering and analysis at scale is something that is, it's growing, it's happening. We see that in certain startups like Recursion, for example. They're doing a lot of this visually with conformal microscopy and so on. But the ability to do this using robotics and imaging tools really will generate huge streams of new data, which will be incredibly useful to us. In addition to that, things like new sequencing tools. But there's a challenge to that as well, which is that, the costs still go up. As a startup, we have very limited funds, of course. Most of that funding needs to go to paying salaries. We pay for compute cost. There are some programs that help, like the NVIDIA Inception program, for example, which can dramatically reduce the cost of renting hardware and or buying hardware and getting cloud access, things like that. But for things like sequencing, we really need to bring down costs still, even though costs have dropped to the point where they're, a few $100s sometimes now. There are things that are maybe still challenging, like single cell attack-seq, proteomics, and so on, which we need to bring down by another order of magnitude in order to really do this at scale across lots of patients, across specific patient populations, and so on.
Thank you. It looks like we have a question from the audience in the chat. And are you all seeing that?
Yeah.
Yeah. Elizabeth, do you want to start?
Sure. This question's like right on the money, right? Like, so if we think about when I started grad school with mass spectrometry, we were probing a cell in 4 hours. And at the first chapter of my thesis was, can we analyze a cell per second? And now we're at like 30 cells a second, right? And like, that's incredible. And that lets you look at like millimeters of tissue, but that's all static. And we know cells talk to one another, and some of those like chemical interactions are incredibly fast. When you add enzymes to the mix, like those are things you can't picture. And so that's why I love some of these other technologies like Raman spectroscopy, because you can actually look at the chemistry in real time in living systems. And so I think like taking videos of that, and I think AI is going to be even critical, because just thinking about how much data we produce in a static tissue, thinking about it in a video context over the course of days to study like development, that's going to be critical. And I think that's the next real omics space. And I think one of the biggest hurdles speaks to cost, right? If you want to do spatial transcriptomics, we're still at like $10,000 a sample. And that's just for like one time point. So I think these two things are going to be running in parallel of how do we deal with that kind of data can we use and leverage some technologies that exist, like spectroscopy, that can look at living systems? And then can we complement that with like mass spectrometry or something that gives you more chemical fidelity? And then can we make it cheap such that you can take videos for days or months or even like I don't know, 6 months, like, and actually capture some of these dynamic systems of especially disease and development?
Oh, thank you. Olga, do you have thoughts on it?
I mean, I completely agree that dynamics is going to be a really important next variable that needs to be tackled. I think some of it, I completely agree with Elizabeth, some of it will be based on really, videos and microscopy, dynamic microscopy of cells, but a lot of it, I think, will also be complemented by basically taking dynamical measurements that are more accessible. It also, of course, partly it's cost, partly it's also just accessibility of data, especially as we think about not just model systems, but also humans, right? Like there's just, practical and ethical considerations. But I do think as we develop and, there's more and more actually efforts really trying to collect different aspects of multi-omic data and trying to put dynamics on this. The other aspect of this, I'll just throw in, I know the question was more about the dynamics, but, the other aspect of this is the vast majority of work right now is really sort of focused on proteins and RNA and sort of going towards proteins. And there's huge explosion now going on in metabolomics, for example, and connecting across of those biomolecules, right? That's going to be another huge, I think, aspect of really revolution, what will bring around a revolution in our understanding. So I think as these projects that are gathering different aspects of the system and probing the system in different ways are taking off more and more, we'll be able to really understand sort of, I'll use the term biomarker loosely, but, what are, for example, mass spectroscopy biomarkers or blood-based or urine measurements that we can use that will give us a good hint at, processes that we can't easily probe in specific tissues and cell types.
Thank you. Neil, what are your thoughts on this one?
Yeah. So I think this is a really great question, but I will keep my reply brief because first of all, there's a lot of complexity here, but also just in the interest of time. Gathering data has two problems, right? So first, there's the actual getting the right data, getting at the right time resolution, and so on. And the next problem, and kind of actually being able to capture it. And the problem after that is really how do you analyze that data? I think there are a lot of insights we can bring to bear on in analysis of data that's like very detailed time points that are happening second by second. But, I think that there are major challenges in actually capturing all this data and capturing it at scale. I used to work in finance and financial services trading systems. You can imagine that when you're doing high-speed trading, there's a similar problem. And one of the key lessons that you take away from that is that as you start to do things in real time, start to react and understand what's happening, the faster the data comes in and the more data you bring in, the less intelligence you can bring to bear on what you're seeing. And so we have to be able to develop algorithms and capabilities that can make kind of major interesting connections and insights at very, very high speeds with not much compute capacity so that we can actually link together the truly interesting things that are happening. I think the biggest challenges are really like, how can you take in huge amounts of data, compress it, save the right amounts of things, and really react within a reasonable amount of time. Even if you're not doing it real time, still there's a time component that goes into analyzing things. And so that's a big challenge.
Thank you. I wanted to also address the issue of models. So Olga, we talked a little bit about this before, about foundational models, purpose-built models. Will we ever really get our hands around biology, or is it just too complicated? I'd love for you to express your thoughts on that. And I found it interesting.
Thanks, Laura. So as you can probably guess from this question, I have very strong thoughts on that. I do think my definition of foundation models, take it or leave it, is not just LLMs, which of course are wonderful, but it's really any model that takes in large amounts of more general data that then can be fine-tuned for specific questions, which of course is extremely useful for biology, because in biology we have a lot, no, not as much as in some other non-medicine related field, but quite a bit of data. But for any very specific question, we often have very limited data. So for example, we have quite a bit of data in what genes are turned on and off in the brain. But if you're thinking about studying neurodevelopmental disorders, you really need data for very early fetal as well as early infancy, and we have very little data there, right? So you can imagine a foundation model could be a very powerful way to do this. I think that it is really important to consider whether, we would ever have, or at least in the near future, have a foundation model trained for all of biology. And my guess is, while, if we define all of biology as something that will help students learn biology, sure, that's great. But if we're thinking about really models that are going to help us really in the precision health and precision medical context, I'm going to bet on really purpose-built models that are going to be really built specifically for medical clinical questions, and perhaps even more specifically trained or at least fine-tuned for specific aspects of the field. And that's what we've seen actually be, the big successes. If you think, for example, AlphaFold, we've all heard of AlphaFold, right? AlphaFold is a wonderful framework, but I would argue that one of its main reasons for success, in addition to the deep learning power, is that it was done with a lot of knowledge of protein structure, right? They're not just throwing in random information from the web and, coming up with protein structures. And, if you look in weather, similar, like very specific domain, the big success of deep learning and foundation models has been coming from models that used a lot of the previous, dynamical weather modeling as part of the input. So I do think that we don't want to basically throw the baby out with the bathwater. We really need to realize that there's a lot we know about biology. It's a very complex field, medicine as well. And so I think that foundation models are going to be transformative, pun somewhat intended. I think they're going to be transformative by really building purpose-built, doesn't mean for any tiny specific question, but purpose-built models in the space.
Thank you. And Neil, I wanted to ask you about, as a company, this, we used to talk about, are you a platform company or a therapeutics company? And you had some interesting insights on how investor expectations are changing in this area. And I think we have many entrepreneurs on the call here I would love to get your thoughts on that.
Of course. Thanks for the question, Laura. So, I think just a few years ago, platform companies were doing, pretty well, raising a lot of money. They were going public, and the expectation was these modern new AI techniques are really going to transform modern drug discovery. But just a few short years later, there's been a rout in the biotech space. I mean, this has been a devastating year, or, the last year was a devastating year, as many people know, for raising funds. Many publicly traded biotechs were being held for less than their actual cash on hand, which means that basically investors expected that they would be destroying cash. So that's a terrible situation to be in from a fundraising perspective. And, to address your question specifically, Laura, I think some of that is because there's this shift, a little bit of a ping-ponging back and forth between what's the value of a platform versus what's the value of individual molecules and therapeutics. I think that it will always be important in biotech and pharma for you to have molecules, treatments that are going to end up going to patients. But then the concept of how do you continue to develop those and how do you create a pipeline, it really varies somewhat based on people's time horizon. And that time horizon, as we know, it shifts with the shifts in the yield curve. So as people become more short-sighted, maybe platforms become less important. As people become a little bit more thinking long term, then platforms become something that they really think about. For us, we thought from the beginning we need to have that pipeline and we need to have therapeutics, which is one reason we became a clinical stage company. So we have 2 compounds, one of which I think I may have mentioned, we recently reported pretty interesting results in a rare disease population, neuropsychiatric results. And those you can find those on our website if you're interested. But the fact that we have these therapeutics available as sort of validation of our platform has also made it hard to raise capital, because people are looking at the therapeutic only, and then they're not attributing any value to the platform. I think as maybe as the economy gets better and people start to shift again to thinking about that platform, then maybe that will shift. But it's really a balancing act, and it requires a lot of kind of dynamic insight into the market and really figuring out where things are going.
Thank you. Okay, in our last 5 minutes, I'd like to talk a little bit about how you approach cross-disciplinary collaboration. It's always a challenge to people speak different languages, right, in computer science and in biology. And so I'd love to hear how you deal with that. I'll start with Elizabeth and maybe quickly go through you guys to close. Yeah.
As a chemist that does a lot of biology, I've navigated this all the time. And I think part of it is if you can get a room of people that put their egos down and are okay to define all the acronyms they're using. Like, I went to a genomics conference, and I think I was the only speaker that actually defined what the technology I was using was. And so just being mindful that maybe when you're trying to translate, not everyone knows what these like alphabet soup acronyms are. And I think that's the first step. And being okay defining that. I also train my students. I make them from a very early start run meetings with physicians. And the meeting starts, and it goes horribly the first time usually, because my students learning that, in fact, they need to take the time to say, okay, this is what our technology is, this is what it gives, but also listen to the physician and do your homework and see, okay, what is this disease? What are the gold standards of studying this disease? And then how do you actually translate that back and forth? So I think it's an aspect of being mindful and then being open and being willing to take that 5 minutes to actually talk about what these acronyms are, what the basis of these things are. I also teach a lot, and that kind of, I think, has helped, because if anyone's tried to teach 100 undergrads and keep them awake for an hour, crossing these different disciplinaries are very similar. You got to keep them entertained. And then I'll stop there.
Thank you. Olga.
So I really think I completely agree with everything Elizabeth said. I also think that this is really the age of collaborative and integrative frameworks. If we're talking about AI in medicine, it's not anything you can do in your corner office by yourself. So I think that it's really not a how to do it or what should we do it, but it's like we must do it. And I think it's actually almost, my group is interdisciplinary. We have computer scientists, we have computational biologists. Pretty much everything we do, we collaborate with clinicians and experimentalists. We actually have an experimentalist in the group. But beyond that, what really has been interesting is starting this Princeton Precision Health Initiative, where we're really trying to change health policy and clinical practice through AI and data science. And that I actually call it transdisciplinary, right? Because it basically forces groups like mine that are already often interdisciplinary to integrate really, because we're trying to really go from genomic and molecular health signals to environmental data, epidemiological level, socioeconomic, lifestyle, social determinants of health. And of course, to do this right, you have to do it in a very explicit ethical framework. So now, not simply are we talking to experimentalistic clinicians and other computer scientists who have never talked to experimentalistic clinicians are now talking to them, but we're talking to ethicists, philosophers, psychologists, sociologists, economists. We have somebody coming in from the Department of Music who's looking at the effect of music in health, right? And so I think it's really critical that you have to be patient and you have to listen and you have to realize that each of us has to change. It's not just about understanding, it's also about if you really want to make a huge difference, you have to be willing to change your direction a little bit and be a little bit more fluid and dynamic, perhaps a lot of bit more fluid and dynamic, to be able to really think about what is the big goal, how can we make the most transformative impact.
Thank you. And Neil, we got one more minute.
Yeah, exactly. When we talk about cross-disciplinary collaboration, as a computer scientist, especially if you've done this for a while, you realize that you have to kind of develop things across many different orders of magnitude. We look at things, sometimes the phrase is from sand to Tetris or something. There are courses for that kind of thing, right? You have to identify foundational bits and then reason from first principles, really understanding like how do things work at this silicon level, the operating system, database, middleware, networking, routing, business logic. And it can span something like 17 to 20 different layers. I think this applies well to biology and sort of regulatory systems as well, because, the computer science mindset really helps you understand that there are a lot of interacting layers, and those layers are not very clear sometimes. However, as a company, we also have challenges collectively connecting these things across multiple domains. Our engineers, they look at things from an engineering point of view, and they're brilliant, right? But we also have to connect that to clinical development. Our engineers, they have specializations ranging from things like particle physics to molecular modeling to pathway biology, and with AI knowledge kind of overlaid on top of that, but they can't be experts at everything. And we also, on the other side, we have experts in clinical development who know the ins and outs of turning a new chemical entity into a drug that's actually useful for patients, by passing FDA regulatory approval, manufacturing it, getting it commercialized, and so on. But connecting those groups, the engineering side and the clinical side and so on, is challenging, and we have to do that across many different levels of abstraction across different domains, and that requires really a huge investment in communication. So
Thank you. Okay, we are out of time. This was wonderful. Thank you to our panelists.